Hey everyone, and welcome to today's lecture on methods in protein biochemistry. So, the first thing I want to take a look at with you is protein purification. And when you want to purify proteins, you basically first have to do cell fractionation, which you see demonstrated over here. And basically, what this is doing is the cells are broken open, and then the cell extracts, which we call homogenates, are basically produced from that. And when you are looking at these, so you see the homogeneous mix of components, and you have that after the cells have been broken open. You then want to be able to centrifuge it. And so you see here the centrifuge, which always make sure to balance, please. When you put a tube of liquid, let's say on the top side here, across from it, you want to put a tube of liquid of the same amount over here, the same type of liquid, same amount to balance it out. Otherwise, oh my God, the sound it makes and the way that you can break that machine. But anyway, you then can do this centrifugation to increase the concentration and the purity of whatever protein that you are that you're looking at. Now, when you are doing this process, I want to point out that ultimately the way that you're preparing that homogeneous mix or that cell suspension is you can either use mechanical or enzymes, enzymatic ways to break down the tissues. And so that's shown over here, how you're mincing the tissues. And you can basically, once you are looking to have that cell suspension homogenize, you can either use sonication, you can use what's called shearing, or you can incubate with mild detergents. And that will ultimately give you the cell extract that you're looking for. Now, I had mentioned a centrifuge and centrifugation. I just want to show you what happens, the order of subcellular fractions when you are using a centrifuge to kind of concentrate and purify out certain fractions from that cell extract. So what you see here is based on, sorry, let me get the laser pointer, based on the amount of time and the speed at which you're running that centrifuge, you can collect different fractions. And you'll notice that the larger and more dense will sediment first. And so, like for instance, over here, you see the nuclear fraction will be first. That's the largest and most dense of the subcellular fractions. Then, if you continue on with that cell extract centrifugation, the next one that you will collect is the mitochondrial fraction, then the membrane fraction. And you'll notice once you've centrifuged down to the membrane fraction, which is pelleted over here, when you're spinning in that centrifuge for that long, the liquid on top, the supernatant, we call that. So you have the pellet at the bottom, and the supernatant is the liquid on top. When you now transfer the supernatant to another tube, that supernatant is now the cytosol fraction. Okay, so what you did first, just to recap, is you had the cell extract, you centrifuged it or spun it down for a little bit. The pellet at the bottom, the first one is the nuclear fraction. You then take the supernatant or the liquid, put it into a new tube, spin that down for longer, and then you'll notice that pellet is now the mitochondrial matrix fraction, and then that supernatant that you put into the next tube, when you spin that down longer and faster, you now have the membrane fraction as the pellet, and the supernatant or the liquid that you remove now is the cytosol fraction. So you have 4 fractions shown here in total. Now, once you've done that, the next thing that you can do is called salting out. So basically, you're going to use differences in solubility of the target protein relative to other proteins in that fraction that you collected. So basically, what you end up doing is you add increasing amounts of very saturated salt solutions to your protein sample. And because of the hydrophobicity of the proteins and the sample, when you have high concentrations of added salt, you then end up lowering the solubility of macromolecules because they're now competing for the liquid solvent there. And one of the things that we use for salting out is ammonium sulfate. And you're going to see that spelt out on the chalkboard notes on the next slide. Now, once you have done this and you've used the ammonium sulfate to salt out your target protein, the problem is you don't want your target protein. You're not going to want to use that protein in experiments while it still has the ammonium sulfate. So then the next step that you would do is dialysis in order to remove the ammonium sulfate. Now, when we're talking about protein techniques, I also want to show you column chromatography techniques. And there are a few different examples for this. There are even more than we're going to go through in this lecture that you'll see in some of your other classes. The first one I want to point out is gel filtration chromatography. And basically, with this, you are going to separate proteins based on their size. So sometimes we also call it size exclusion chromatography. And you see it in the picture here. Basically, you'll have a column that's filled with these matrix beads that are very porous, meaning that they have a bunch of different sized holes in them. Now, when you pour your protein mixture on top to travel through the matrix, when you look at these, beads that are these big ball-like structures with the little holes in them, smaller proteins are going to be able to enter those holes. Now, if the small proteins are entering those holes, it's going to take them a lot longer to travel through that column because picture, I got to go through this bead first, then I got to travel through this bead, then I'm bounced into the next bead, and they're going bead by bead. So small proteins will be very slow to travel through the column, whereas large proteins, what's going to happen to them? They can't fit inside of the beads of the matrix that's filling that column. Instead, they're going to slide right off the outside of those beads and just travel right down the outside of that column. And so large proteins will be the first ones eluted. Eluted simply means what you are pulling out of the solution, okay? So when you look at gel filtration, or when we call it size exclusion chromatography, gel filtration chromatography, the first proteins to exit will be the largest. The last ones to exit or elute will be the smallest proteins, okay, because they get trapped inside those beads and take a long time to come down. Now, sometimes for gel filtration chromatography, you will also see the term HPLC. That is a high resolution version of basically, this gravity-based gel filtration chromatography that will have a column matrix with, extra small beads in it, much smaller pores and matrix within it, and high pressure, that's what the HP is for. High pressure will be used to force the buffer and the proteins through the column. And so with HPLC, because the beads, the pores and the holes are so small, and because you're using high pressure, you end up getting greater separation of proteins, meaning that you're able to separate proteins that are really close in size to each other, whereas in regular gel filtration, if you have, two different small proteins, they might not separate out from each other much because, the beads are, how small can the pores really be? One small protein will fit through the same small hole as another small protein. So HPLC lets you separate a little better, but it's also going to be a lot more expensive and more technical to use. Okay, so gel filtration, also known as size exclusion, which includes also HPLC, all of those, we were using size of the proteins to separate out proteins within a protein mixture. This one here that we're looking at is ion exchange chromatography, and whenever you see ions, that means there's a charge involved. So this is for exploiting the charge differences between proteins and to separate proteins within a mixture based on which protein has a positive charge or which has a negative charge. Okay, and so you have two different types of columns that you can do for this. You can either have a column that is positively charged. So the resin has a positive charge on it to then interact or bind negatively charged proteins. Or you can use a column with a matrix that has negative charges on it. Okay. And so that will basically be for binding um, positively charged proteins. And so they basically name them based on what they're trying to attract. So a positive charge resin is going to attract negative proteins, and we call negative charged proteins anions. You can underline the N in anionic to remind you that's negative. And so this one here would be called anion exchange matrix. You'll see it written out on the next slide, which is DEAE. Uh, and with that, then the other one, if it is negative charge on the resin, it's going to bind positive proteins, and positive we call cation. So that's a cation exchange matrix, a CMC. And I always use the trick cations think of a cat. And, they're cute and positive, unless you're allergic. Then again, I am so sorry for using that analogy. But you'll notice that you have the charge on the beads, and that will attract the particular protein. And then they use an elution buffer to saturate those beads and elute out, remove the currently bound protein. The next one is affinity column chromatography. And with affinity chromatography, whenever you see affinity, it's basically how attracted to something, is how much it loves it, wants to bind to it, and whatever. It's kind of affinity. Things with a high affinity for something will want to stick to it, will be, you know, following it around, kind of like students with me. They're high affinity for me. I can't get rid of them. They are stuck to me. So affinity relies on the binding or attraction properties of the target protein. And so with this, you basically will have a high affinity ligand for the target protein linked to the matrix bead. Okay, so over here in white, you see the bead itself. And then you'll notice that the little gray circles stuck to that bead are the ligand covalently bound. And that's a ligand that the target protein likes to bind to. Okay, so the target protein needs to have a high affinity for that ligand that's coating the beads. Okay, all the other proteins in the mixture, the non-specific proteins, they will fall right down the column. They're not going to be attracted to that ligand. They don't want to bind to it. So they slide right down. They come out first. And then what you do after is you elute with competing ligand to saturate the beads and the ligand. So basically you have the target proteins pulled off of the beads. And when we look at affinity chromatography, what you can also use this for is you can have an antibody column that can be used to isolate antigenic proteins too. And we'll talk about that in later slides and later lectures as well. So now, when you have purified a protein, meaning you've purified your target protein from a cell mixture that had, the homogenate had a whole bunch of different proteins, at the end of that, you want to know how efficient was my purification? Did I get a good job? How pure is my sample? And I know this slide is scary because it's got a lot of numbers and it's going to have calculations, but I will put on the next slide the chalkboard notes. You'll see exactly how to do each of the calculations. So make sure that you know how to calculate each of the things we're going to go over on this slide. The first thing I want to show you is specific activity. You can determine specific activity by dividing the total units of activity by total protein. So over here we have total protein. Here we have total units of activity. So for each of your different samples, the protein that was extracted from these different techniques, basically, you would be able to take a look over here. You would be able to calculate the specific activity by dividing the total units of activity by the total protein. So the purification of our target protein by preparation of a crude cell extract, our specific activity here is 10 because you take 20,000 and divide it by 2,000. Okay, here 36 is because it's 18,000 divided by 500. Okay, and so on and so forth. The next thing that you can calculate is the multifold increase in purity. And the way that you get that is you divide specific activity at any given step. So any of these steps, you divide specific activity um, by the specific activity at a previous step. So that's why you notice that there's no multifold increase in purity for the first step, because you need to have two steps to compare. So this is the first step, that's the second step. At the first step, there's no previous step to compare. Okay, at this point, what you can now do is divide the specific activity at that step, which is 36, divided by the specific activity of the previous step, which is 10. Okay, so 36 divided by 10, you get 3.6. Over here, when we want multifold increase of the third step, we take the third step, divide it by the second step, and that gives us 2.5. Okay, and you could do that for any of these. There's going to be a practice at the end that I'll have you send me in the Remind app to make sure that this is clear to you. The next one is total yield percent. The way that you get this, and now keep in mind it says percent, so that's why you're going to have to multiply by 100 at the end. So the total yield percent, you divide total units of activity at a given step by the initial units of activity, okay? And then multiply by 100 to get a percentage. So total yield, again, is gotten by dividing the total units of activity at that step. So total units of activity is this column here. Total units here for this step is 20,000 divided by initial units of activity. Okay, and so in this case, the initial units of activity would be, well, the first units of activity, which is right here. And then you multiply by 100 to get the percentage, okay? Now, in this case, because it's the first step, if you're taking 20,000 divided by 20,000 is 1, and you're multiplying by 100, that's why you have 100 here. For the second step here, the way that you would calculate it, again, we do total units of activity, 18,000, but this time we're dividing by the initial units of activity, which is the first step here, 20,000. And now you multiply that by 100, okay? For this third one, this number here, total units of activity at this step, divided by the initial total units of activity. So for this one, we're always using that first total units of activity as our denominator. And don't forget, you have to multiply it by 100 to get it to be a percentage. The next one and last one that I want to show you is the total multifold purification. Okay, so that you do by dividing specific activity at a given step by initial specific activity. Okay, so again, whenever we say initial, that means we're looking at the number that's at the first step. Okay, so over here, let's look at, let's say, the third one. If we wanted to calculate the step 3 total multifold purification, we would divide the specific activity at that given step, which over here is 89. You divide it by the initial specific activity, which is 10, the one that's at the very first step, okay? And that gives you, over here, 8.9. Here you would do 289 divided by 10. Here you would do 767 divided by 10. Okay, now if any of these steps were a little confusing to you, I'll be able to help you further when you send me the remind question that you're going to find at the end of this lecture. Okay, now the next slide will give you a recap of each of the calculations that we just went through. The next method in protein biochemistry that I want to go over is gel electrophoresis. And gel electrophoresis is an important technique not just for proteins, but also for things like DNA. So you've probably worked with it in some of your other classes and laboratories, including intro bio labs, and if you took genetics already. The one we're going to focus on today, though, it's for proteins, and basically it's a way for us to approximate the molecular mass of a protein, and also to kind of figure out if after you've done, let's say, a protein purification, you want to see, did you get your target protein? Is it the only thing that you got in that sample? Running a gel afterwards will help you figure that kind of information out. Now, the way that this works is you end up using what's called polyacrylamide gel electrophoresis. So when it's for proteins instead of DNA, DNA will use an agarose gel and you'll see it flat on the table. Whereas for protein gels, you'll see them standing up and they'll be called PAGE for polyacrylamide gel electrophoresis. And it's going to separate out the proteins in a sample based on charge and size. Okay, so basically, the way that it's going to do separation based on charge is you'll look, one side of the gel has the cathode, the negative, the other side has the anode, the positive, and you use sometimes in the gel, in the buffer as well, you end up using what's called SDS, sodium dodecyl sulfate, and that will coat the outside of your denatured proteins. Now notice they're denatured ahead of time to help you run them through the sample. So, the buffer that you have them in, you will be using to denature them. The SDS will also help denature them as well. It's going to be important because it's coating them with a negative charge. Okay, so the purpose of SDS is to be a detergent that adds a net negative charge to the protein. That's important because when you load them on the gel, you're putting them in the wells that are near the cathode, which is the negative side. Because you have now coated them in a negative charge, it's going to make them want to repel away from this top portion of the gel down toward the positive side, the anode. Okay, and so they will want to run toward that positive charge because they're currently at a negative charge. Okay, now In addition to the proteins running across the gel because of charge, the other aspect of this that's important is that whenever you have gel matrices or gel matrix, whether it's agarose for DNA or polyacrylamide for the proteins, you can't see, but there's these tiny pores that make up that matrix. It's almost like when we talked about the beads in size exclusion chromatography. So now what happens is as the proteins are running across through that matrix of the gel, the larger proteins are going to get stuck in that porous gel closer to the well. They're not going to be able to travel very far. Whereas the smaller the protein is, the easier it'll be able to navigate through those microscopic pores that we can't see, and it will run further down the gel. So further away from the well is going to be the smallest proteins, and then closer to the well is going to be the largest. The way that I always tell it to students to give you a visual to imagine is picture a jungle gym. Okay. Little kids can run through that jungle gym like it's nothing. They could get through the slides, the tunnels, everything because they're so tiny. If an adult, one of their parents, tries to chase after them, what's going to happen? They try to go through that same tunnel or that same slide. They're going to get stuck because they're too big. Okay, so that's kind of what happens in terms of the gel. Okay, so please remember the largest proteins will be close to the well, the smallest proteins will be furthest away, and they will be toward they'll be traveling toward the positive charge on the other side. Okay, so just remember, page is separating based on charge and size. And you can use different gels. Basically, the density of that gel, or what we call percentage of polyacrylamide, will help you to basically separate different size proteins as well. So you can make a gel with a higher percentage, lower percentage that will adjust which proteins can travel through the pores, because that will adjust basically, how dense the gelatinous gel is and how far, how the proteins can migrate through. So that will be valuable for you if you want to separate proteins that are closer in size to each other. You can modify the percentage of the gel. Okay. Now, once you run the gel, these are nice little pictures of it. You can see all of these bands as different proteins that are present, and you'll know the target protein based on the way the sample has been prepared and based on the marker that you always load. Now, when you're analyzing gels, I want to kind of point out a few different things. First of all, whether it is a DNA gel or a protein gel, anytime you run a gel, please make sure to always run what we call a marker. Sometimes we call it a ladder or a standard, okay? And that's critical. The marker or the ladder is basically a commercial tube that they sell that will have a given amount of known protein fragments. And it's important that they're known because it means that you know the exact sizes of the bands that will appear. So the bands are the lines. The exact sizes of each of those bands you know because that is a sample with known fragments of protein. And that's very important for you to then use to compare to any of your sample lanes. And you'll be able to say, okay, my target protein is around 121 kilodaltons. So if my marker has the 121 kilodalton line over here, in my sample lanes, I'm going to be looking in that area. Okay? And that's always very important to have. Now, what you can do if you want to analyze an unknown protein in your sample, what you can do is you use that marker to make a graph of the standards. And basically you graph the known mass of each of the fragments compared to the distance that it migrated or traveled on your gel. So the distance away from the top wells. And you plot each of those standards, each of the known proteins you plot on a graph comparing the mass to the distance traveled. Once you have that standard curve, you can then take the unknown protein band, wherever, whichever one you're looking at, and you can plot the distance that it traveled on your gel. And from that, you can estimate what the mass of that unknown protein is. Okay, so you can, use that plot and see where your unknown protein falls on the ladder, because sometimes you're analyzing a sample and you don't know the details of the protein that you're looking at. Now, there are different versions of gel electrophoresis. It could get a bit fancier and kind of try to give you more information. And so the ones that we were looking at a moment ago are called one-dimensional SDS page, and those are the ones that are going to separate, the proteins on the basis of the different sizes, and they're able to run across that gel based on the charge. Now, we also have what's called 2D gel electrophoresis. So instead of those single, lanes of lines, you end up getting this crazy, what it looks almost like a scatter plot of specks everywhere. And this is basically because this one will first separate the proteins on the basis of what's called the isoelectric point. Okay. And then, so from low to high, PI means the isoelectric point. And then it will further separate on the basis of size, just the way the one-dimensional gel did as well. Okay, we're not going to go into crazy details about the technique of this one or anything. I just wanted you to know that it exists. It's very valuable for us because the purpose is that it can allow us to resolve like 1,000 to 2,000 separate proteins in one gel, basically, in one technique. So it gives you a lot greater resolving power than the 2D, than the one dimensional. Okay. And just in case you ever see the term over here, DIGE, that just stands for 2D differential in gel electrophoresis. Okay, but again, we're not going to go into the details of these. I just wanted you to know that this exists and that it gives you a greater resolving power than one-dimensional gels, and for you to know that the reason that is it's combining not just mass but also isoelectric point analysis. Another valuable technique in analyzing proteins is called sequencing, so that you can determine the sequence of the amino acids making up that protein. Now, it looks crazy and scary, but I'm going to show you a kind of quick and easy way to do sequencing, for instance, Edman sequencing. When you are looking to do Edman sequencing, you're going to have your polypeptide that's been digested with trypsin, and then the same polypeptide digested with chymotrypsin. Okay, and so you'll have different fragments for each of these digestions. Now, what you first do is look at your fragments between these two and ask yourself which of these fragments over here starts the same way as one of the fragments over here. So when look at this, VPD, VPD over here. So this will be the first fragment that you write out for yourself. That tells you that's the beginning fragment because they both start the same out of all of these different fragments, okay? So you line them up over here. Then you look at the overhang. So notice they're the same, they're the same, they're the same. And then the chymotrypsin one ends, but the trypsin one over here continues on. So now we're going to look, we want to find, okay, well, this one ended. Where does it now continue? Well, over here, the next letter should be R. So let's go to our chymotrypsin fragments over here and find that overhang, which is just the letter R right now. Well, here we have R. Okay, so we know that the next fragment of chymotrypsin should be RICTTL. It's this fragment here. Once we've placed that fragment, well, we now have a different overhang. Our trypsin ended at the letter R, right? Well, now we have ICTTL that our trypsin does not have written out yet. So now we go back to trypsin, and you're going to keep alternating back and forth. So now let's go back to trypsin. Let's find that overhang piece, ICTTL. Look at that, it's right over here. Okay, so now you would draw or write that out over here. Well, look at that. Once we do that, we now have a new overhang to look for. Let's go back to chymotrypsin now, right? Because we've ended over here at chymotrypsin, we need to find the next fragment. So our overhang right now is NS-IgGr. So where is NS-IgGr out of our chymo fragments? Right here. So now we would write this whole fragment next in terms of our chymotrypsin. Okay, chymotrypsin over here. Now we have another overhang, right? We have this ASW. Well, let's find that ASW on our trypsin part right up here. Okay, so you would write out that next portion, and that ends up giving you the end of the fragment. So you see, you then write out the whole fragment that you've made listed out. It sounds a little confusing when we're looking at it this way. I'm going to give you an example that we're going to go over together to show you how we can do this again from scratch. Okay, so that was a lot of information at once. So to make it a little clearer and make sure you understood what was going on in the previous slides, we're going to go step by step through an actual problem for sequencing the protein with Edman degradation. So this is a practice problem from one of Dr. Shen's previous exams. Like I mentioned before, you would have a bunch of fragments from digesting your protein with trypsin and a bunch of fragments from digesting your protein with chymotrypsin. So now what you would do is look at your trypsin fragments, okay? We want to see if any of your chymotrypsin fragments start the same way as one of your trypsin ones. So let's look at our first fragment. Starts with a Q. Do any of these chymotrypsins start with a Q? No. How about a C? Let's look down here. This one starts with a C, but let's see, does it match this one? This is CQGL. This is CCXF, so that's not our start. This third one, AND. Let's look here. Oh, look, we have AND over here. So then what we would do is we would take this and say that's the start of our protein sequence, AND. So now let's see. We write that over here, our trypsin fragment, okay? ANDCR. Now we take our chymotrypsin fragment, the start of that one, and we write it down here. Now, when we look at these, we now have an overhang present. So we say, okay, this is in our blue fragments here. So we want to find which red fragment fills in that overhang. So we have the overhang of QEGF. Let's go to the red fragments. Where do we have starting QEGF? Right here, this one right here. So we would write that as the next red section, right? So we filled in that overhang. Now our overhang is IIHK. And so I recommend whenever you're doing these, it really helps if you have two different colors or some way to distinguish the two so you know which fragments you're going to look at. So we have red IIHK. So we need to find the blue fragment for IIHK, and look, it's right here. So now we can fill in the chymotrypsin and find what our next overhang is. So now when we write out this whole fragment here for chymotrypsin, now we go back to our trypsin and say, okay, what trypsin fragment starts with PMTSW? Which trypsin starts with PMTSW right here? So we would now fill in that next segment. Okay, now what is our overhang? VBANDR. Okay, we are up to blue, right? Where the overhang shows that we're missing part of our blue sequence. So let's go to the blue fragments. VBANDR is right here. So we would write that whole fragment, which is VBANDR QQY. So we write that here. Now what's our overhang? QQY. So we need to fill in the red QQY, which is right here. Okay, so we would write this whole fragment right where that overhang is. Okay, now we have a nice long overhang fragment to look for in the blue section. So we're going to look for CCX in the blue fragments. Okay, so what do you notice? This little CCXF, we fill that in there. We still are missing some blue fragment, right? So we need to find the rest of the blue fragment over here. So let's go back to the blue fragments and find which one starts with XZVBR. XZVBR right here. So let's fill that in over here. Okay. Now, what is our overhang that we have to find? We have to find CQGL. Which fragment starts with that overhang? CQGL right here. So you would write that whole fragment out down here. Now our overhang, which fragment starts with that overhang? HMM SSK. So let's look at our fragments. What starts with HMM SSK right here? Right here. And it looks like we're pretty much getting to the end. Okay, so now the only overhang that we're now missing is that red tanks, I like to call it, TANX. Okay, and now you'll notice we have now used up all of our fragments here. So 6 red ones and 6 blue ones, we used them all up. So now we can write out our whole sequence. And the trypsin sequence in red should be exactly the same order as the chymotrypsin in blue because you were sequencing the same protein. You were just doing a different digest. So you would go step by step and let's see what our final sequence is. This is the order from here to here. So you would write, first we have the ANDCR, then we have QEGF, and you would keep writing each of these letters in order. Okay, and now the next one is right here. Okay, so you would just follow and see what order you have everything in. Okay, and like I said, you could also use the chymotrypsin order. That's the same exact thing. The blue, even though the fragments are different lengths and different starting points, it's all the same sequence. So notice when we write that out, then we continue here, it's all the same sequence, okay? If you have any trouble with this, just contact me in the Remind app and I will help you. We can do another practice problem as well. Again, just send me a picture of whatever slide's giving you trouble and send it to me in the Remind app. Okay, sorry about that. That was a lot of information. You may want to take a quick snack break or TV break or something. Recommend Supernatural um, before you continue on because that sequencing always drives students crazy. Um, so now, moving on to some other protein-related techniques, we have mass spec now. So mass spec, it basically allows us to measure the mass of small peptide fragments. And what exactly it's measuring is the mass to charge ratio. And ultimately, this is a great way for us to identify proteins based on their mass. And there's also a version of it called tandem mass spectra mass spectrometry. And for that one, that's what you see here. The very first step is electro spray ionization. With tandem mass spec, you're basically using it to predict the amino acid composition of trypsin or tryptic fragments. And basically, you put those fragments into the mass spec with them going through the electrospray ionization first. And what that basically is high voltage that is applied to a liquid to create an aerosol. And aerosols, most students know now, thanks to the pandemic, what an aerosol is. But basically, you end up with this spray of ions that can then run through the rest of the machine, and you end up having these spectrum analyses that are possible to identify the protein based on mass. Okay, now we're not going to go into crazy details of all of this because I just want to introduce you to a bunch of different techniques, and then in other courses and whatnot, or other lectures within this course as well, we can go into more details later on. The next technique that I want to briefly mention is called solid phase peptide synthesis. And basically, what I just want you to know for this is that they use this to synthesize antigens, or peptide fragments for antibody production. And antibody production is very valuable in biology and chemistry and any of the sciences, very valuable to the research world and the medical world. It can also be used to test biological activity and kind of look at, substrates for reactions with amino acids that aren't kind of the natural ones that you would see. But again, I just want you to know this is a way solid phase peptide synthesis, which is shown here, can be used to synthesize antigens for antibody production. And it basically is an automated way to generate polypeptides, usually of around like 25 amino acid residues. We call it kind of quick, but it's within a few hours. And it's basically a 5-step procedure that you see mapped out for you here. Now, in addition to all of the stuff we've been talking about so far, like sequencing and whatnot, you have to remember that proteins are not just the sequence of their amino acids. A big aspect of proteins is how their structure is ultimately arranged, and things like folding and whatnot. That's what we always say in biology and in biochem and chemistry is structure defines function. Now, two very powerful, valuable ways to look at protein structure are X-ray crystallography and what we call NMR. So NMR stands for nuclear magnetic resonance spectroscopy. I always mess up that word. X-ray crystallography, what this is basically relying on is the diffraction of X-rays by protein crystals. And then NMR is exploiting magnetic properties of atoms. Now, the big difference is that NMR is capable of revealing dynamic or changing structure of a protein, whereas X-ray crystallography, that's only going to give you a static model, a static kind of structure analysis. Now, a little while ago, we mentioned how we can use certain techniques to develop antigens for producing antibodies. And I said that antibodies are really valuable to us in the science and medical field. And that's emphasized here. Some of the terminology I want you to know associated with antibodies. First up, we have epitopes. And basically, an epitope is the specific site on an antigen that can bind to the antibody. And when I say antigen, remember antigen molecules are like a name tag protein. For instance, people now know the antigen known as the spike protein on COVID viruses. And when it comes to antigens, they can have multiple epitopes, and each can be specifically recognized by a different antibody. Okay. Now, when it comes to antibodies and epitopes, you're going to see two terms. You're going to see the term polyclonal antibodies and monoclonal antibodies. And the figures here, you don't have to memorize them or anything. This is just to show you in case you're wondering how they produce polyclonal, which is up here, this one, and monoclonal, which is down here. And I'll never forget back when I was doing my PhD research in Dr. Shen's lab, we had to develop, we had to use one of the companies to have them make for us a polyclonal antibody. And they actually give you the schedule of like when they're preparing each step. You see the immunization over a 6-week period and all. And they then tell you when they are killing the bunny, and they killed the bunny on my birthday. And I didn't know any of that was going to happen. And then I felt horrible. And then it was even worse when that antibody ended up not usable toward our project. But we're not going to go down that road of horrible memories that occur when you are in the sciences. But yes, please go toward the sciences, because in the end, it is the most fun area to be in, despite traumatizing moments like that little bunny moment. So now, the difference between polyclonal and monoclonal, when you look at their words, poly versus mono. Poly means many, mono means one. So if you have a polyclonal antibody, that's going to be able to recognize multiple epitopes of an antigenic protein. Whereas monoclonal antibodies, mono, that antibody will recognize one and only one specific epitope on an antigen. Okay, and then in the lab courses, when you take classes like micro and whatnot, we go into the details of when and why you would want to use the two different types. But for now, polyclonal, know that that means the antibody is going to recognize multiple epitopes on an antigen, and monoclonal, it will only recognize one epitope. Then we get to Western blotting. And Western blotting, even though the textbook puts it at this point in the lecture, you actually have this connected to gel electrophoresis. And Western blotting is something that's very valuable in all of the different classes in the sciences that you take and all the different research that you can do. Western blotting, the purpose of this is to identify proteins that have been separated by the SDS PAGE gels that we mentioned earlier in this lecture. And basically, you end up using antigen specific antibodies to identify the exact proteins that have been spread out on that gel. Now, what you do with this is the main steps. You have that gel that you ran. You then transfer it to a special filter membrane. Usually it's called nitrocellulose filter. And that's basically specifically has pores that stick, that protein stick to. You would then wash it and do things like, block the rest of the spots where the proteins aren't, so that you don't get non-specific binding. You would incubate it with a primary antibody, then a secondary antibody, then visualize it. Now, what I want to emphasize, rather than, we're not going to go through in detail all the steps and memorize all of that, the most important thing for you to know right now is the difference between the primary antibody that you soak that membrane with and the secondary antibody, because this comes up a lot in all different biology and chemistry experiments and procedures and all. So when you hear primary antibody, that is a specific antibody that's protein specific. For instance, if you are looking for a tau protein, tau being related to Alzheimer's research, you would have an antibody that specifically binds to tau and only tau. The secondary antibody, on the other hand, that is a non-specific or general antibody that will bind to any of the primary antibodies. Okay, so that one's not binding to tau in our example. Instead, that one is binding to the primary antibody. What's special about the secondary antibody is not only do I want you to know that it binds to the primary antibody, but it's covalently linked to an enzyme that produces a detectable product. What that means is the secondary antibody binds to the primary antibody, and the secondary antibody has a form of detection on it. So, for instance, it might have something that fluoresces that you can detect in the next steps of your experiment. Okay, so the secondary antibody is actually what allows us to ultimately see the target protein on the membrane. Now, sometimes you're doing experiments or you're looking at proteins that have not been thoroughly studied by other researchers, meaning that there may not be an antibody to target that particular protein, to pull it down, to visualize it in a Western blot, for instance. And so what they do is they came up with procedures to epitope tag proteins, meaning to take any protein you want and add a specific little epitope sequence, so a sequence of a few amino acids, okay, that you can stick on to that protein, right? And once you add that to that protein, there are commercially available antibodies to target it. So now, even though, let's say, protein A that you're studying is not very well known and there's no antibodies that you can buy for it, all you do is you put that little flag tag or MIC tag right here. You put that on your protein, and now you just buy the already made anti-flag antibody, and you can use immunoprecipitation to pull down your protein from a mixed sample, or you can use the antibody to visualize your protein on, let's say, a Western blot. Okay? So, very valuable to us. So, epitope tagging is when you put a small amino acid sequence onto your protein. Immunoprecipitation is when you pull down proteins from a mixed sample and you use antibodies to be able to pull down your target protein from a mixed sample and collect just that particular protein. Okay, there are just a couple more techniques that I want to briefly mention again, just so that you're aware that they exist, but we go into more detail in other lectures and in other classes. This one here is immunofluorescence. This allows us to identify proteins that are inside cells that have been chemically treated in a way that does not damage the rest of the cell architecture. Okay, and it's also antibody based, and so it allows you to be highly specific when you are looking for target proteins. And basically, after you have prepared and washed your cells, you can visualize the protein that you're looking to study. You can visualize it through fluorescent microscopy. And basically, you would be using the concept of primary antibodies and secondary antibodies again to have fluorescence in here. And so you could end up with some beautiful images. But the difference with this compared to some of the other techniques that we talked about is you get to actually visualize your target protein in a cell structure. Okay, the cell has not been damaged. Whereas a lot of the other techniques we talked about, you cracked open that cell, you burst it open, and you harvested just the protein. And then the last technique that I want to mention today is one near and dear to my heart because it relates to microbiology, which is one of my favorite classes to teach, along with genetics as well. And so this is ELISA, and ELISA stands for Enzyme Linked Immunosorbent Assay. And this is very valuable to us. It's a technique that allows us to detect particular proteins in a sample. It can identify very low level antigenic proteins, and we are able to use it with a lot of different biological samples. The reason why I mentioned microbiology is that ELISA is one of the big methods that we use for diagnostics. So, for instance, if you take microbiology classes at CSI, you will do an experiment, a practice ELISA, where you are looking for HIV in samples, and you can detect which patient samples are infected with HIV and which ones are not. So you're able to look for a particular antigen in a sample through using a series of antibodies. And again, it's the same thing we mentioned with Western blot, in that you would be able to have a primary antibody specific to the antigen you're looking for. And then you have a secondary antibody. So in this image, here's the primary. You have the secondary antibody that has a form of detection on it that can then do something like change color. So only the wells that have the antigen you're looking for will change color, whereas the negative ones will not. And one other thing that I want to mention, I could talk for ages on ELISA, but for our purposes in biochem, we'll be brief. One other thing that I want to mention is, in addition to this being used in diagnostics, like some of the COVID tests that you may have taken, another very famous ELISA is for what I call the parasitic infection that you are stuck with for life, aka pregnancy. So, pregnancy tests are a form, an everyday form of ELISA. So, circle star highlight that in your notes because I like the fact that with a lot of the techniques that we talk about, like NMR and mass spec, these are things that students would not have necessarily encountered in their everyday life. And yet with ELISA, every time you walk through a Walgreens, a CVS, or maybe in your own personal experiences, you've all heard of and seen pregnancy tests. So I like to highlight that for you to think of ELISA. Also, COVID tests. There were a lot of ELISA-based COVID tests as well. Okay, so before I let you leave for the day, here is the practice problem that I would like you to take a picture of and send me your answer in the Remind app. I want you to fill in all of these empty rectangles based on the notes that I gave you earlier in today's lecture. And if you're having any trouble with that, again, these are things that I don't want you to cheat on, take someone else's answers. There's no point. You're not graded for whether you get it right or wrong. You're just graded for having sent it to me. And it lets me help you. So if you look at this and you're like, oh my God, I had no idea what you were talking about, which I know I get that a lot. All you would have to do is send a picture of this and say, I am so confused. Can you help get me started? Or if you think you want to try it on your own first, send me a picture of your answers and I will let you know if you are on the right track. And that is it for today. I'm sorry, this was a kind of exhausting and long lecture because it did include some calculations and problem solving. Again, any slides that you have any trouble with, feel free to send me a picture in the Remind app or write your questions as comments on the YouTube videos. I am happy to help. Thank you and have a great day.